really the fundamental cause of the disease and the understanding of what happens inside our cells is not information that we can easily access with MRI and also the other modalities, despite the fact that the magnetic resonance is inherently extremely well suited to provide this type of information, because we have the spectral information, the spectrum that provides information about the molecular structures that are involved. And this is something that we use in NMR spectroscopy, but which is also possible to do in vivo with MR spectroscopy. However, due to the very low sensitivity of magnetic resonance, it's extremely difficult to detect these very low levels of endogenous metabolites. So we can do proton-based MR spectroscopy with coarse resolution, long averaging times, and this provides some useful clinical information in some circumstances, but is not very widely used clinically. And if we infuse traces or contrast agents, metabolic substrates, and try to follow their fate in the body, then we really struggle with signal to noise. As you can see from the example on the right, after infusion of very large quantities of C13 enriched glucose, we can get almost no spatial resolution, but just a spectrum from the entire brain, or a very large part of the brain, and we have very poor spatial resolution. But we know from these NMR spectra that we can have a lot of information about what goes on at the biochemical level. So this is really the background for what we set out to rectify. And one of the techniques that we pursued was the hyperpolarized, the dissolution dynamic nuclear polarization. We also worked with parahydrogen. As many of you probably know, we had a very strong focus on that. Oscar Axelsson, Helge Johannesson, with colleagues and collaborators, developed parahydrogen for exactly the same purpose. But over the years, it turned out that dissolution dNP was a very excellent technique for providing hyperpolarized biological substrates that were of interest. So the idea with dissolution dynamic nuclear polarization is that you can take your biomolecule, formulate it with an electron spin. It could be an organic free radical. You put it in a high magnetic field, cool it to a low temperature, liquid helium temperature, so around 1 to 2 Kelvin. And then you irradiate it with microwave close to the resonance frequency of the electron spin, and the electron spin will polarize the nuclear spins in its vicinity. And by dNP, we can then enhance the nuclear polarization to tens of %. We then do a very fast phase transition from the solid state at low temperature to the liquid state at room temperature by dissolution. So it's injecting a hot solvent buffer to dissolve the bioprobe, the biological molecule. And then we end up with a liquid solution of the agent that we can then measure in our NMR spectrometer or inject into the patient. dNP has the disadvantage that it's quite complicated compared to what you do with PHIP, which is extremely elegant in many ways. I've always been very envious on the PHIP experiment, the shake and bake approach. With dNP, we use much more complicated equipment. We need a superconducting magnet because we need a high magnetic field. We need low temperatures, so we need cryogens, liquid helium. We need large mechanical pumps to reduce the temperature of the helium bath. And we need microwave sources to re-rate the electron spin. And then finally, we need what is shown here as a dissolution stick. We need to inject the hot solvent into the cryostat to dissolve the frozen biomolecule to get the liquid solution. So the instrumentation is expensive, complicated, and challenging to maintain. And here you see a video of how one of the first prototypes actually worked. So this is at Oxford University. One of the first prototypes we built, the sample has been sitting in the superconducting magnet for an hour to be polarized, and now it's ready to be dissolved. The two PhD students put in the stick to inject the hot solvent and receive the hyperpolarized solution, can quickly run to the MR scanner and inject the hyperpolarized solution into the tail vein of the rat, in this case, which sits in the MR scanner with a coil on the chest above the heart, and you see the hyperpolarized pyruvate, in this case, substrate, and you see all the metabolites being generated in the myocardium of this rat. So this is what the DNP experiment is, and since, of course, this complicated instrumentation can be made more practical, which is one of the key focus areas for us, one of the targets, that this has to be packaged in a way that it's acceptable in the clinical environment, that can be done by a technician, a radiographer, lab technician, and not two trained PhD students, and it's safe to inject it into the patient. So here you see an overview of the development of dissolution DNP instrumentation. The first prototypes were the basis for the Hypersense product from Oxford Instruments, which was served the in vitro field. It's now discontinued, and a new let's say preclinical in vitro polarizer is available from the company Polarize, which is coming out of my lab and my work. But also the initial polarizers were used in the first clinical trials. So we took one of the first prototypes, put it in a clean room at UCSF, and that was the basis for the first patient study. GE, in parallel with that, developed the SpinLab, which is the commercial polarizer, which is what is currently the only practical solution for clinical hyperpolarized MR. So the SpinLab generates the hyperpolarized solution. The operator takes the MedRat syringe from the quality assurance, quality control system. It's being released, and then the operator quickly runs to the MR scanner and injects it into the patient, and then the imaging takes place within the first minute during the lifetime of the hyperpolarized state, the T1 of the hyperpolarized agent. So the signal from the hyperpolarized solution, of course, decays very quickly. It's being diluted in the bloodstream. It relaxes. So the hundreds of millimolar that we can inject of the contrast agent, quickly, the signal quickly decays as we get to the millimolar concentrations in the tissue, and as time progresses due to T1, which is typically on the order of a minute. So we are of course interested in selecting nuclei that are magnetic, that have a magnetic resonance signal, obviously. Nuclei that have long T1s. So the low gamma nuclei are of course preferred, spin 1/2. And then we need to label molecules in a suitable position. So for biological work, the obvious candidates are of course carbon-13, nitrogen-15, possibly phosphorus. Some other nuclei could be of interest in exogenous molecules that have specific contrast mechanisms. And the agents that we target are energy metabolites of the energy, let's say, synthesis or the generation of energy within the cells. So small metabolites that have fast motion and therefore long relaxation times, and also labeling positions that have long T1, so like carbonyls and carboxylic acids. And of course, there are a number of biological requirements that I'll get back to in a minute when we look at one of the key examples of a very favorable contrast agent, Pyruvate, which is the top of this table. But these biological substrates, of course, have to have very high uptake into the cells. So transporters into the cells have to be very efficient, and also the enzymatic pathways that we are targeting also have to be very efficient. So it is a very, let's say, demanding biological experiment. It's an experiment which has many constraints, and therefore we have to be clever in terms of selecting the substrates that are of potential interest for hyperpolarized metabolic MR. And since even though, despite the strong enhancement that we can create, we are still quite insensitive, and therefore we also need to inject very high doses of the contrast agent. And I think this is, I really think that this is quite a fundamental limitation of magnetic resonance, is whether independent of which technique we use for hyperpolarizing the biological molecule, that we still need to give fairly high doses. And many of the agents that you saw in the table before, I mean, both whether they're endogenous or exogenous contrast agents, the safety profile is in a favorable case could typically be on the order of 0.1 millimole per kilogram body weight. That is the approved dose level for a gadolinium contrast agent, which is eliminated renally by the body. So endogenous molecules which have no pharmacological effects would probably have safety levels on that order, or maybe possibly a little bit better. But that also means that we have to hyperpolarize a fairly large amount, like 10 millimole for a typical patient, or 1 to 2 grams of the substrate of the imaging agent. And with DNP, we have other constraints that we also need to formulate it in a way that we can add the radical, and that we have a good glass, and it stays amorphous as we freeze it in the polarizer and do the DNP. And therefore, we need additives of some kind that is also biological accepted. So this is, of course, another general constraint that we have to remember when we pursue specific substrates, whether it's one or the other technique, that the final formulation that has to be given to the patient has to be well tolerated, have a high safety profile. It is a diagnostic contrast agent. We're not curing the patient. It's not therapeutic. It's diagnostic, and therefore it has to have a very high safety profile and be eliminated in an acceptable way with minimum side effects. And with DNP, there are many different ways of formulating the samples. Some are very straightforward, like pyruvate, pyruvic acid, ethyl pyruvic acid, ketoisopropylic acid. Some of the exogenous agents that we have synthesized, that they form good glasses as neat agents. Others are salts and have to be formulated in a way that they have high solubility. We minimize the amount of co-solvent. It could be cesium salts, tris salts, large counter ions that increase the solubility. Or we could go to acidic or caustic preparations of for instance, amino acids that also improve the solubility of these amino acids. But this is a constraint that we have with dissolution DNP, that we have to formulate it in a very high concentration, the imaging agent. So what you saw in the video was an illustration of what the dissolution DNP is, how it takes place. But of course, as I said, in a clinical environment, it has to be automated and packaged in a completely different way, both for patient safety and from the operator perspective. So the SpinLab, which is developed by GE, serves that purpose. It's fully automated. It has closed cycle cryogenics, which means it doesn't consume any liquid helium, but still reaches a very low temperature. It has multi-sample polarization because, as I said, DNP is a fairly slow technique, so the sample typically sits in the cold environment for an hour. In this case, it has 4 independent channels, and we need to ensure that what we inject into the patient is sterile and safe to inject, and that is done by the fluid path, which is sterile cassette, where we can load the contrast agent in a pharmacy, in a clean room, where we can ensure that the sterility of the product. And then, of course, we need a large dose, so patient dose, as I just explained. And finally, before the product is injected into the patient, we need to ensure that everything has gone well. So we do some QC before the product is released, and that also has to take place within seconds, because time is of the essence, and the T1 is short, and the clock ticks. So the SpinLab is very successful in the sense that it's capable of reaching a very low temperature without consumption of liquid helium. It actually reaches 0.8 Kelvin for a long time, but it uses what we call the absorption pump, so it has to be sort of regenerated overnight. And that's the way it conserves the helium, that it just absorbs as a pump the helium vapor and reduces the temperature. And then during nighttime, that helium can be regenerated, desorbed from the absorption pump and recondensed by a cold head. One of the key, let's say, inventions with the SpinLab was what we call the fluid path. So that's basically adopting concepts from nuclear medicine, where these sterile single-use cassettes are also very often used to contain the chemicals of the synthesizer for generating the radioligand. Here, there's no chemistry involved, but still, this was the idea that was adopted, to use these disposable cassettes. It's a little complicated and expensive for disposable, but it does serve the basic purpose that it contains. It can be loaded in the pharmacy in a clean room under sterile conditions, aseptic conditions, and then the sterility is maintained into the polarizer and throughout the process until it's released and injected into the patient. So you see here a sample vial, which you can see on the left photo that it contains about 1 to 2 grams of the contrast agent that can slide into the cold region, into the polarizer through this dynamic seal that you see on the right photo. And then we have a dissolution syringe which contains the solvent that dissolves at the end of the polarization process, the imaging agent. It then goes through some tubing further to a receiver, and before that, what is called here on the photo, the EPA filter, is the filter that removes the radical, the electron spin, because now it has served its purpose. It has helped us with the dDNP, the hyperpolarization, but there's no reason to inject that into the patient, so it's filtered off. And then the receiver has a number of QC appendages, which are basically just optical cells that allows us by optical means to measure that all the parameters are correct, and the process has gone well and it's safe to inject. I think I'll just quickly run over these because I can see I'm a little bit behind on time, but everything is automated. All the insertion of the sample, the heating of the solvent, all is done in an automated way. And I hope you can see here from this video that everything now goes in a very expedient, automated way. You can hear the sound from the cold head on the SpinLab and the compressor, so that's what provides the cooling capacity. You can see the 4 tubes behind the door. One of them is now being dissolved. You can see the tubing flicker a bit. That's because the hot solvent is injected. And then the sample can be simply retrieved. This is a video that is taken without the quality control system. I'll show that in a minute, but you can see here the hyperpolarized solution. And here you see a video of the quality control system. So the liquid that was retrieved manually in the previous video is here shown as it's being retrieved in the quality control system. The video in the center is just looping, so you see it many times, but you can see that the receiver has some solvent. You can see that the hyperpolarized solution enters the receiver and fills it up as a clear liquid because the green radical has been removed, and then it's pushed from the receiver into the MedRat syringe on the right. And in between those two, all the quality control has been done, and all the parameters that you see in the table on the lower right have been measured. So it's being checked that the pyruvate concentration is within range, that the radical has been removed, pH is correct, temperature is correct, volume is correct, and also that the polarization is fine. If the polarization is not correct, then there's no reason to inject it into the patient, even though it would have been safe. But all the others are part of the let's say process and product control that ensure that the that it's safe to inject into the patient. Okay, this was a bit about the instrumentation behind the solution dDNP. As I've been talking already about Pyruvate, which is the substrate that has translated to the clinic today, the only product or the only molecule that is clinical today. And Pyruvate is very central in energy metabolism. It's the end product of glycolysis when glucose is broken down, and just as it enters into the Krebs cycle. So pyruvate has several fates. It can be reduced to lactate by lactate dehydrogenase. It can be transaminated to an amino acid like alanine, or it can lose its C13 label as CO2 if it goes into the Krebs cycle. So it really reports on several pivotal pathways within the biochemistry of energy metabolism. Therefore, it's also a good probe of what we call the Barbarous effect. You can see here an example in a tumor model where a rat has a tumor inoculated on the stomach. You see the metabolic images on the lower left as pyruvate has been injected. There's a high uptake of pyruvate into the tumor, and there's a lactate hotspot because a lot of the pyruvate has been reduced to lactate. And that is really a demonstration of the Barbarous effect, that tumors have increased energy metabolism, aerobic glycolysis, as it was demonstrated by Barbarous. And it does really tell us a lot about or characterizes the cancer in many ways, which I'll give you a couple of examples of from a clinical point of view as the final couple of slides in my talk. So this is data from the first patient study that was done at UCSF. You see here a patient with confirmed prostate cancer. So you see the anatomical proton image in the center. You see the dark area in the prostate. Well, firstly, maybe I should describe the very dark area at the bottom is the rectum where the coil sits. And just above that, you see the center of the image, which is the walnut-sized shaped prostate, and you see the dark area within that, which is from the anatomical image already the location of known to be the location of the tumor. And here you see that in this case, this patient received a pyruvate injection, and this was just done without any spatial encoding, except for 2 slabs being acquired. You see the dynamic traces on the lower left and right from a slab through the normal prostate and one through the slab with the tumor. And you see the timing of the bolus arrival, which is extremely fast. So it's IV injection, and already less than 30 seconds after the IV injection, the pyruvate reaches the prostate. And when and in the case of the slab through the tumor, we see lactate being generated with a few seconds delay from the arrival of the pyruvate bolus. This is extremely encouraging because it tells us not only what we knew, that the pharmacodynamics, the arrival of the pyruvate, but also that the tumor has a very high uptake of the pyruvate and generates enough lactate that we can actually image that. So this is what we see in the next example of another patient, where you also see from the proton image the dark area, which is the tumor, the known tumor. This patient is now being imaged with an EPSI sequence, and we can then generate an overlay lactate over pyruvate map, as it was done back in those days. You see the lactate hotspot in the tumor area, just as we had hoped and expected. But in this case, for this patient, it actually turned out that the patient had bilateral cancer, which was not known. So the patient, actually it had an effect for this patient, even though it was just a safety study. These men are on active surveillance. And in this case, due to the bilateral cancer, this patient went off active surveillance and had the prostate removed. So it really shows a very nice example that shows us that we need better tools, that we couldn't see everything from the anatomical proton images and all the other diagnostic workup that we have available. It's really a very difficult decision for the medical doctor to decide on whether this patient stays on active surveillance or goes for some more radical treatment. And actually, UCSF have gone on to look at other patient populations, and also continued with the prostate cancer indication, trying to stage the prostate by correlating the pyruvate over lactate, or KPL, the rate constant of pyruvate to lactate, with the grade of the tumor, and also looking at response to therapy, as in this example, where a patient that went on to that was castrated at hormone therapy, that actually we can see that after the treatment with the chemotherapy and the androgen deprivation therapy, the hormone therapy, that the lactate signal completely disappears, and actually it's a very good nice demonstration that we are able to pick up responders from non-responders, and that you can see that this patient actually responded to the treatment, which is also extremely important, because we know that in cancer one of the challenges is that with anatomical imaging, you have to wait many weeks and have to go through many cycles of treatment that may not be efficient before we can actually see a physical change or size change of the tumor. So having a method that can early tell us whether the treatment is effective is extremely important for the outcome of the individual patient. We can either switch to a more effective treatment or continue with the treatment that we know works. So I hope I have given you an overview of how dissolution DNP works, that you agree with me that we have, since we work with magnetic resonance, we have many advantages over other imaging modalities. We have an non-ionizing radiation modality. We have a lot of information in the MR spectrum that provides us unique information about biochemical pathways. We've come a long way. We have the first clinical trials. We made the clinical translation. For dissolution DNP, one of the challenges is to make the polarizer technology simplified and cost-effective. I'm sure that there are equal challenges with PHIP, even though the technique inherently has many advantages and is much more simple in many ways. But I think some of the learnings that we have and some of the requirements that we have had with dissolution DNP will be similar for translating PHIP into clinical applications. So thank you very much for your attention, and I'm happy to take questions.
Thank you very much, Jan. That was fantastic. As you might expect, we have several questions, so I'm going to start reading some for you.
Sure.
First, it's a two-part question. To what degree is the amount of pyruvate that you're injecting in these human subjects limited by the biological tolerability, or is it limited by what the device can produce? And secondly, how does the large bolus affect the metabolism?
Well, I would say it's entirely limited by safety. We could produce a larger dose, but the 0.1 millimole per kilogram was defined by the safety study, the tolerability of pyruvate. It is a fairly high dose that is given as a bolus injection. So there's some acute effects that we have to take into consideration. There could be some pharmacological effects of pyruvate. We have not seen them, but they're reported. It's also been used as a therapeutic. So it might, if we went to a higher dose, that might also be effects. Right, that it is if you consider like the let's say the pyruvate concentration in the bloodstream or in cells, it is what we could call a super physiological dose. We are let's say giving the cells a fairly high dose, but I would say that at the clinical dose level, the 0.1 millimole per kilogram, with the transporters, natural transporters, enzyme activities, we're not saturating enzymes, and I don't think that we we're perturbing the actual metabolism to a large extent. So I think the question alludes to the possibility that we could be introducing an artifact because of the high dose that we are giving. But I think all the preclinical and clinical data that we have, such that we are actually measuring in a, let's say, a rigorous way, the Warburg effect.
All right, next question. Practical one, how is the radical filtered out?
In the video I showed you, it's simply by precipitation. So it's we just do an aqueous dissolution of the pyruvic acid, so it stays acidic, and the radical, the green powder, just precipitates out and is removed by mechanical filtration. It can also be done by reverse phase resin. So if we did a neutralizing dissolution, or if after, I mean, in a second step, we could also push it through a reverse phase filter and remove the radical that way. So there are several methods.
All right.
Currently clinically.
How rapidly is pyruvate taken up into cells after injection?
Yeah, that's exactly, well, how should I quantify that? I would say extremely fast. I cannot give you a number in terms of rate constant, but as we could see from the dynamic curves on the patient, we don't know exactly whether it's what's in the bloodstream, what's inside the cells. It's only the lactate signal, which has to be produced by the cells and possibly is then excreted from the cells into the bloodstream. But we can see that at the clinical dose level, as soon as the bolus arrives at the prostate, we see by a few seconds delay, we see a strong lactate signal. I think that if probably the question, maybe I can qualify it in a sense, define the number of steps and that we has to be coenzymes present. So there are many factors that play into the effective KPL or rate constant that we could model from a kinetic modeling. The monocarboxylic transporters can be a limiting factor. So transport is a key factor. And of course, that's also regulated by the cells and in different diseases, the transporters. So it is important, and people have done studies of inhibitors of transporters. So roughly, and of course, as we go to very high doses, we will also see some saturation of the transporters and enzymes. So I would say it's fast. That's a short answer. It's pretty fast, but we have to be very selective because that's not the general situation. It is one of the challenges in picking the right substrate is really that where can we find substrates that have high uptake and fast enzymes in the cells, and that's really the challenge.
All right, two last questions. The first last question is, are the patients fasted before pyruvate, and does that matter? And then, so I'll ask you that question first.
No, I don't think so. I have to confess, I cannot say with 100% certainty with the different protocols and how it's set up at different institutions, because by today there are 8-10 places in the world that can do studies, and they have many protocols approved by the FDA. They with 100% certainty in general, but no, they are not. And you could say that's typically what's done with FDG and in nuclear medicine with PET. But I don't think there's any proof that it actually has an effect with pyruvate.
Okay, and lastly, given where things are now, what do you see as the biggest barrier towards translation into the clinic or further study by clinician scientists with this technique?
If I should answer specifically for dissolution DNP, then, well, we are already in the clinic. We have these 8-10 sites that have patients, and they have many studies funded and approved. I think there are 24, 26 studies approved. If you go to clinicaltrials.gov, you will see them, different patient populations. So we're already there. But I think really to go from where we are today, the big challenge for us is to define what we would call the killer application. It's really important for full clinical development for any company to register this as a product, that there's a good business case. And what we need to find, which has been the challenges for gases in many different areas, really to find the clinical application indication where we can make a difference for many patients that has enough good enough business for registration and the millions of dollars that have to go into clinical trials.
All right. Well, thank you very much for a fantastic talk again, and for stepping in under such short notice. We really appreciate it.